I'm a biology professor here, and if you're interested, or if you know somebody that's interested in giving a talk about some scientific expertise, you can contact me or really anybody in the biological sciences or physical sciences department. So we're always looking for somebody to come and share some scientific knowledge with us. So, one other quick note before we introduce Josie is that we have one more science talk this semester, Tuesday, March 31st. It's at 11:30 a.m., so earlier in the day. And GRCC professor, geology professor Taria Maddox is going to discuss volcanic eruptions in Iceland and Hawaii. And the title of her talk is A Tale of Two Hotspots, Fire and Ice. So anyway, put that in your calendar, right? All right, so I'm here to introduce Josie Mitchell. And Josie grew up in the Upper Peninsula of Michigan in Sault Ste. Marie. Earned a Bachelor of Science in Chemistry from GVSU while also playing on GVSU's varsity volleyball team. Currently, Josie is a third-year PhD candidate at the University of Wisconsin-Madison in the integrated program of biochemistry. She works in Jill Waldonger's lab. Did I say that last name?
Yeah, pretty close, Wildonger.
Pretty close, sorry. A lab where she's studying the biochemistry of how neurons develop in the fruit fly model organism. And they use techniques including CRISPR, immunohistochemistry, and live animal imaging with a confocal microscope to study the biochemistry and cellular biology of a neuronal ion channel that allows the fly to sense pain. And obviously she's here today to discuss some of that research with us. So if you would, please give Josie a warm welcome and thank her for coming here today.
Thank you. Thank you, Tim. All right, thank you guys. Thanks for coming and sticking around to the end of the workday and coming to my talk, and thanks for the opportunity to give the talk today. So I'm going to tell you about how organisms feel pain, and I'm going to tell you what we can learn from the fruit fly model organism on this topic. And so before I get into the meat of my talk, I'm going to introduce myself a little bit. I went to a virology seminar in grad school, and at the beginning of that talk, every presenter would give a little background on how they got to where they are. So my story centers around the Midwest. I'm a Michigan girl. I grew up in the Upper Peninsula of Michigan in Sault Ste. Marie, and it was here that I asked my first scientific question. And my first scientific question was, how do pea plants germinate? So that was for the 4th grade science fair here. And then for the 5th grade science fair, I asked my second scientific question, which is, how do horses see color, or do they see color? And this was a really fun project for me because I also rode horses when I was a kid too. So after high school, I graduated and went down to GVSU, and I got my bachelor's in chemistry there, and I played college volleyball at Grand Valley. I also got into undergraduate research there after my freshman year. So I joined a lab where we were studying antibiotic resistance. So this is how bacteria can destroy antibiotics and become resistant to them. And so we did X-ray crystallography in this lab. And so shown here are pictures of crystals, and these are microscopic crystals of that beta-lactamase protein. And so the work I did in that undergraduate research was then published, and we published a couple of crystal structures in that paper. So then after undergrad, I decided to go to grad school. So I took a hop across Lake Michigan, and now I'm in Madison, Wisconsin. And so in Madison, I'm studying fruit flies. And I'm not just studying the fruit fly as an animal, but I'm actually studying something inside of them called a neuron. And this is a picture of a neuron inside of the fruit fly. And I'm here in Madison with a really great cohort of other grad students, and also my husband Josh, who's sitting in the back today. And we all work on this campus in Madison. So we're surrounded by 2 lakes in Madison, and this is the biochemistry complex right here. I think my lab is right about here in the complex. So Madison's been a really great place to live and go to graduate school. So if anyone has questions about my path to grad school or what grad school is like, please feel free to ask me those types of questions too. All right, so now to get into the meat of the talk, and I'm actually gonna switch my microphone because I keep turning my head. So to get into the meat of the talk, all organisms have 5 main senses. And these senses are vision. And vision can allow us to do things like cross the road and avoid cars and traffic and getting hit. We also have hearing, and hearing allows us to hear things like an ambulance siren or somebody talking to us. We also have our sense of smell. So we've all been here before, probably. But smell can alert us to something that's dangerous, like fire or smoke. We also have our sense of taste. So this allows us to enjoy this brownie sundae, for example. And then the final sensation, which I'm going to focus on the most today, is the sensation of touch. And touch can be good. It can be a nice sensation, like a hug from a loved one. But it can also be bad in the form of pain. So I've been here a few times growing up on a farm in the UP. But pain is something that we need to be able to sense in order to survive. So organisms that can't feel pain oftentimes don't survive very long because they can't protect themselves. And so the big picture question of my research is, how do organisms perceive harmful environmental stimuli? So how do they sense that? And to talk through this, I'm going to go through a couple of points. So first, I'm going to talk a little bit about how sensory neurons communicate painful stimuli. Next, I'm going to tell you some about how model organisms have helped us learn how pain is transduced in sensory neurons. And then finally, I'm going to talk about my own research on a pain-sensing ion channel in fruit fly sensory neurons. So first, how do sensory neurons communicate painful stimuli? And what exactly is a sensory neuron? Well, to get to this point, I want to tell you a little bit about what pain is in the first place. And there was a really nice issue in National Geographic this last month. So back in January, this is the cover of the National Geographic, and they were highlighting pain as a critical health concern. And so there was a great graphic in here. And so if we think about the example of hitting your finger with a hammer, if we zoom into this graphic, the very first line of defense against that hit with the hammer comes from sensory neurons that are embedded in our skin. So these sensory neurons have to be able to take that stimulus and convert it into something that the brain can understand. And so they do that and send the signal along their axons all the way up into the brain. The brain can process that and then retract the hand from that painful stimulus. And so this process is happening at a speed of 200 miles per hour. So from the hammer hitting the finger to the signal getting sent to the brain, it's instant. So this process has to be like hooked up and happening very fast. And so how do neurons take this signal, like the hammer hitting your finger, and change it into a signal that the brain can understand? Well, there are ion channels, and these ion channels are like little doorways into the cell. And those little doorways allow ions. You may have heard of sodium, calcium, potassium. They're all charged ions, and they can then pass into the cell and carry their charge to make an electrical charge in the cell that can be sent as a signal. So we're going to talk a lot more about ion channels. But just remember that ion channels are part of these sensory neurons, and that the sensory neurons send the signals to the brain. And so back to kind of the basic question, we're looking at these sensory neurons, but what exactly is a neuron? So a neuron is a single cell. And just like other cells, neurons have a nucleus, which is housed in the cell body. But this is different than most cells that you might see in a textbook. These types of cells have very specialized compartments. So they have dendrites, which kind of look like branches on a tree. I swear, since I've been doing this research, whenever I look at a tree, I see neurons, basically. So the dendrites are the signal receiving part of the neuron. So they're going to take signal and send it towards the cell body. And then that signal is going to be turned into an action potential that will be sent down the axon, and then eventually it's going to be sent to the brain. So through this electrical wiring of the body, the signals move in this direction. And so we have all sorts of neurons in our body, but the neuron that I'm studying is a sensory neuron. And sensory neurons are going to perceive environmental stimuli. So they can perceive signals such as mechanical pain, thermal pain, or chemical pain. So mechanical would be like that hammer hitting your finger, or a sharp poke or a pinch. Thermal would be like burning your hand on a pot. Or chemical would be something like a chemical burn. And so the body has to be able to take many different stimuli, even the good pain, or not good pain, but good touch, like a hug, and be able to send those signals to the brain and have the brain understand. And so how are these signals sensed? Well, you might remember from the National Geographic slide that there are ion channels. And these ion channels are located in the dendrites. And remember, the dendrites are receiving signals. And so these ion channels are going to let ions flow through just like a doorway. And if we think about these like doorways, there's a unique doorway for every different kind of ion. So if we've got different ions like sodium, calcium, and potassium, this door is only going to let, for example, sodium go through. So there's different ions and different ion channels, and they're very specific on what they let through them. Okay, so sensory neurons are going to have these ion channels that allow them to perceive these stimuli. But the big question in our field is, how do these ion channels get to where they need to be? You may notice that these neurons are pretty big cells. They're not the typical like round cell with the nucleus in the middle. These cells are quite specialized, and they have all of these different projections. So these ion channels are actually transported by something called a motor protein. And so these motor proteins are really unique, and they are going to take the ion channel and drive them around the cell. So you can think about this like cars on a highway. So motor proteins are going to drive cargo, and cargo would be something like the ion channel. It might be something like mitochondria, and it's going to drive that to the right location at the right time. So there's a lot of regulation that's going on inside a neuron, a lot of things that have to go right in order for a neuron to work properly. And so to emphasize kind of what this movement is looking like, there's a really great video. And this is a video. It's been loading, but sometimes it takes a little bit. So this is a video of a motor protein. And this may come down to, I've just got not great Wi-Fi signal here. So let's see.
You want me to log you into the internet here?
I did log in already. All right, well, we'll try to play that after because I think it's really worth seeing. But what's happening here is this motor protein is walking along this object here, which is like the road in the cell, and it's carrying this big vesicle right here. So the motor protein can carry vesicles with cargo to different locations in the cell. And so one of our questions in our research is, how is this localization of pain-sensing ion channels regulated within sensory neurons or sensory dendrites? So we want to know, how do these things get to the right place at the right time? And if we think about this in kind of a cartoon format, all cells have a cell membrane. They have a nucleus, which contains DNA. And in order for things to be made in the cell, the DNA must turn into RNA, and that RNA has to then turn into protein. And so the protein in this case is our ion channel. But it doesn't just stop here inside of the cell. The important thing is ion channels have to make it to the cell membrane in order to function. So they actually have to be inserted in this membrane in order to feel painful stimuli and then allow ions to pass through. So there are these doorways into the cell. An ion channel inside the cell isn't going to do much good. So we need to know how these ion channels get to the cell membrane. And so in order to look at this question, many scientists have turned to model organisms. So if you think about it, it would be really hard to study pain in a human. It'd be hard to inflict pain ethically in a human and be able to study that. So we actually use different model organisms to understand how pain is transduced in sensory neurons. And so a few examples of model organisms I'm showing here. So model organisms have made really great contributions to science. So there's the fruit fly, which many people are probably familiar with on your kitchen counter, flying around old fruit. There's also the mouse, and there's the worm, C. elegans. There's the zebrafish, and then there is baker's yeast. And so you might be thinking, these look nothing like humans. How can we learn something from an organism so small? But in fact, these organisms actually share quite a bit of DNA with humans. And so I highlighted that here. Drosophila, or fruit flies, actually share 75% of their disease-causing DNA with humans. So something that causes a disease in humans, 75% of that DNA is actually found in the fruit fly. Same thing with the mice. Mice actually share 90% of disease-causing genes with humans. And then worms are 83%, zebrafish are 70%, and yeast is surprisingly 23%. If you think of how simple yeast are and how they can share something with a human, it's pretty incredible. And so what I want to emphasize here is that model organisms have been really helpful for discovering things that then apply to human health. So things that are discovered in an organism can then be applied to learning more about human disease. So for example, the worm, C. elegans, is what I'm going to tell you about in a second. But the worm is one of the first touch sensors that was discovered. And so in the worm, the touch sensors that were discovered were MEC4 and MEC10. And MEC4 and MEC10 make up this ion channel. Remember, it's an ion channel in a neuron that's going to sense touch or pain. So these are mechanosensitive channels, so MEC. So they're sensing that mechanical pain like touch or like the hammer hitting your finger. And so these mechanosensitive channels, MEC4 and MEC10, belong to this larger family called degenerin epithelial sodium channels. And so from that name, can anyone guess what ion these channels are going to pass through? Sodium, yeah. So these ion channels are very specific to sodium ions. And so remember this name because we're going to come back to this family of channels in a little while. So this is the worm touch sensor. What about the mouse? The mouse touch sensor is piezo2. So piezo2 was also recently highlighted in a pretty big journal called Nature. So piezo was highlighted in this article, and this is an X-ray crystal structure of piezo, and this is as if we were looking down from the top onto the ion channel. So if you look at it, this would be the hole where ions would go through this channel. And what's really interesting about piezo is scientists found that when mice don't have piezo, they can't feel a cotton swab stimulus on their paw. And so this black bar is representing mice that don't have the piezo2 protein. And when they don't have that, they don't withdraw their paw when they feel that cotton stimulus like controls do. So this is a normal mouse. This is one that lacks piezo. And so this protein was discovered back in 2010, this piezo protein. And really interestingly, 6 years later, it was actually found in humans. So humans that are lacking piezo also lose their touch perception. So this is a really key example of something that was discovered in a model organism, like a mouse, that was then found to be implicated in something that humans had disease or problems with. And so in this article, in the New England Journal of Medicine, they had 2 patients. Both of them could not feel touch. They also had a lot of problems with their balance and ability to sense their environment around them. Here are 2 of the experiments that they did. So one is a vibration detection experiment, and this red bar is representing those patients, and they couldn't feel vibrations. So about under half or 50% of the stimuli were not felt by these patients. Another experiment they did was a 2-point discrimination test. And if you've ever taken a physiology class, you may have played around with this, but when you poke with 2 probes, you should be able to tell that you're being poked with 2 probes, depending on how far apart they are. But these patients couldn't tell that they were being poked with 2 probes, no matter how far apart they were. So they couldn't discriminate those pokes. So these patients had a problem with their touch sensation, although I want to highlight that both of the patients could still feel touch on their hairy skin, so something like their arm. And this is different because there's different neurons that innervate your hairy skin versus your palm or forearm here where you don't have as much hair. Another thing is these patients could still feel painful and thermal or heat stimuli. So that you might be thinking, well, how do they lose one and they can still feel the other? Well, there's different neurons sensing different things, and then in those neurons, there's actually different ion channels. So different ion channels can feel heat, different ion channels can feel painful stimuli, and so on. So they lack this, but they can still feel a lot of other things. And so now this brings me to my favorite pain-sensing ion channel, and that's pickpocket. So this is the pain-sensing ion channel in fruit flies. And researchers a few years ago discovered that fruit flies that don't have the pickpocket ion channel feel less pain. So this bar is representing animals that have a deletion of that pickpocket gene. This is our control bar. And the experiment that's done is shown in this video here. So basically, in this video, you can see what's a Drosophila larvae. So this is a baby Drosophila. And it's going to be poked with this probe. When it's poked with the probe, it's going to roll into that stimulus. So we can actually do an experiment where we inflict pain on these larvae, and they'll actually roll. So we can measure a response from that painful stimulus. And so what happened in this experiment is that the experimenter would poke the larvae, and 80% of the control larvae would roll, and they could feel the pain. But only about 60% of the larvae lacking pickpocket would roll away from the pain. So this channel, this ion channel, is required for larvae to feel pain. So you might be thinking, why do larvae need to feel pain? It's something I didn't really think about before I was studying this, but larvae actually need to feel pain to be able to protect themselves. And in the wild, they're protecting themselves from something called a parasite wasp. And this is also a video that may or may not play, so we'll give it a second to load. But basically, yep, I don't think we're going to have luck with the video. Basically, the parasite wasp is going to sting the larvae and inject its egg into the larvae. The egg will then consume its host and turn into a wasp. So Drosophila larvae, which are kind of here, and the wasp is attacking one, Drosophila larvae have to be able to feel that painful stimulus and roll away and get away from the wasp. So it's a pretty interesting pain response. And we can actually take that pain response and then study its neurons and figure out what's going on to allow the larvae to feel pain. And so in my research, we used Drosophila a ton as a model system. So has anyone heard of a model system before? Or a model organism? Probably a few people. Okay, so model organisms I've mentioned, and using these model systems, we can actually take these problems like sensing pain, and we can do experiments in the model system and learn what happens there to then apply to humans. So most of us are familiar with this version of the fruit fly, but fruit flies also go through many developmental stages. So they're first laid as an embryo, they then turn into larvae, and the larval stage lasts about 3 days, and then they're going to pupate, where they develop a hard shell around them, and then hatch into adult flies. So this whole cycle takes about 10 days. So fruit fly are a really great model system because they're fast. We can breed these flies, and 10 days later, we have more flies. So you can generate a lot of generations of these fruit flies to do your experiments. It doesn't take as long as a mouse, which has to go through gestation and all of this longer time scale. The other nice thing about fruit flies is that in the larval stage, which is about the size of a grain of rice, the larvae are actually transparent, so they're see-through. So here's a picture of a Drosophila larvae, and it's expressing green fluorescent protein in its sensory neurons. So we can actually image these larvae with a confocal microscope, and we can see into the animal. So unlike something like a fuzzy mouse, you can't see inside of them when they're alive. You'd have to dissect the mouse. But in fruit flies, we can actually visualize things inside of them when they're still alive and intact. So this is really powerful because we can study things that would be normally or physiologically happening. So zooming in on one of these neurons, you'll see that this is one sensory neuron. So all of these tree branches that you see are connected to this cell body and this single axon. So all of these dendrites are sensing signals from the environment and then sending those signals to the fruit fly brain. So we study these sensory neurons, so I look at these about every day under a microscope. But the other thing that we can do in fruit flies is use gene editing techniques. So many people have probably heard of CRISPR. And we can actually use CRISPR in the flies to edit their DNA. So our lab was actually one of the first labs to take CRISPR. So it was discovered back in 2012-ish. And then our lab took CRISPR and brought it into fruit flies for the first time. And so now I've told you a little bit about sensory neurons and how they communicate painful stimuli. I've also told you a little bit about how we can use model organisms to understand pain transduction in sensory neurons. So now I'm going to go into the third part of my talk, which is my own research on a pain-sensing ion channel in these sensory neurons. And so the ion channel I'm going to tell you about is one that I introduced earlier. This is called pickpocket. And pickpocket was discovered back in 1998, so it's really only been known about for about 22 years. However, it took a whole 12 years to figure out what pickpocket was really doing. And so in about 2010, scientists found that pickpocket was required for Drosophila larvae to feel pain. So that video I showed you earlier where they were poking the larvae with that probe, that's the pain response of the larvae, and they found that pickpocket is needed for that pain response. And so what is pickpocket? Well, pickpocket is a pain-sensing ion channel in Drosophila. It belongs to a larger family, and this should look familiar. This is the degenerate epithelial sodium channel family. And what I'm showing here is a large phylogenetic, or basically family tree for these proteins. So in humans, we have the epithelial sodium channels. These are located in human neurons. They're also located in our kidney. So in the nephron of the kidney, they can regulate salt balance. There's also the acid-sensing ion channels, or ASIC channels, and these are involved in a lot of the inflammatory response with pain. So oftentimes with a painful stimuli, there will be an acidic environment in that painful stimuli, and these ASIC, or acid-sensing ion channels, will sense that acid. In C. elegans, I told you about MEC4 and MEC10. So they belong to this family. And then in the fruit flies have 31 ion channels that belong to this family. And so you might be wondering, well, there's pickpocket protein in fruit flies. Is there a pickpocket protein in humans? We haven't found it yet, but the pickpocket protein in fruit flies is very similar in its structure to the proteins in humans. So what we learn about pickpocket in a fruit fly, we can apply to ion channels in humans as well. And so back to my main question, which is how is the localization of ion channels regulated? I'm going to narrow this down even further to the question that we can study in fruit flies, which is how is the localization of pickpocket-1 proteins regulated in sensory dendrites? So basically, how does the ion channel, in this case pickpocket-1, get from being inside the cell where it's made to where it needs to function in the cell membrane? How does this process happen? So in order to be able to study pickpocket, we have to be able to visualize it somehow. So if you just stare at a fruit fly larvae, you don't have laser vision, you can't see pickpocket just with the naked eye. So what we have to do is use markers, and these markers are called antibodies. And what these antibody markers do is they're going to bind to our target, which in this case is the pickpocket protein, and they're going to have a fluorophore, something fluorescent on them, so something that will light up when you shine a laser on it. So this marker is going to label pickpocket protein, and when we zoom in, you can see these arrows pointing to the signal, which is the pickpocket-1 protein. So you'll see that it's kind of all along these little projections in the dendrites. So the black signal is the marker that's labeling this pickpocket protein. So in a control fly, this is what we see. However, in a fruit fly that is a mutant, and so this mutant I'm going to show you is a mutant for the Fragile X protein. So that's the FMR1 protein. When the fruit flies lack this Fragile X protein, they actually don't have as much pickpocket-1 that's inserted into the membrane. So the difference from here to here is in control flies, you see the pickpocket-1 protein, whereas in the FMR1 mutant, there's less pickpocket-1. So the signal is greatly decreased. And so to demonstrate this in more of a picture format, in controls, let's say we've got 5 pickpocket channels in the membrane. When there's no Fragile X protein, we'll say there's a decrease, so there's only about 3 channels in the membrane. Obviously, there's lots more channels, but in this example, there's a decrease in ion channels, and that's shown right here. So with no Fragile X protein, there's less pickpocket in the membrane. And so this was really intriguing preliminary data for us. So all of a sudden, we're seeing this pain-sensing ion channel, pickpocket 1, being regulated by something that is actually pretty critical to human health. So you may have heard of Fragile X syndrome, and this is caused by loss of the Fragile X protein. And so in Fragile X syndrome, this is a genetic condition, and it's caused by changes in the FMR1 gene. And those changes in the gene, or the DNA, are going to cause that protein to not get made. It affects about 1 in 10,000 people, and it tends to affect males more severely because it's an X-linked disease. Additionally, it can cause developmental delays and intellectual disabilities, and it's actually one of the leading genetic causes for autism. So Fragile X syndrome is a pretty significant syndrome that's caused genetically. And one other thing that I want to mention is that it also causes defects in sensory processing. So many people who are affected with Fragile X syndrome have problems with their sensation and sensing their environment. So it's really interesting to us that this pain-sensing ion channel is actually regulated somehow by the Fragile X protein. And so, again, this is causing defects in sensory processing. And so our question now boils down to, how does FMR1 regulate the membrane expression of the pain-sensing ion channel pickpocket 1? And so I mentioned earlier that in order to study pickpocket, we have to be able to see it. And so the way we've been seeing it for years, since its discovery in 1998, is we've been using an antibody. However, and the antibody looks something like this when we are looking at the neurons. However, when we use an antibody, we can't look at a living larvae. We actually have to cut it open. So what I'm showing you here is a larvae before it gets cut. And then if you picture dissecting a frog, you may have done it in middle school or high school, it gets cut down the middle and opened up like this. And so that's shown right here. So this is the larvae that's dissected. And then you apply this antibody marker in order to see these structures. Okay, so this is the way we've been seeing it for like 20 years. But what I told you earlier is that we want to be able to study these processes in a live animal, so something that's not dead. We want to be able to see something that's physiologically relevant. So in order to do that, we want to visualize PPK1 in a live, intact larvae. And we're going to do this using CRISPR to tag PPK1 with GFP, or green fluorescent protein. And so a little bit of a review on what CRISPR is. So CRISPR stands for clustered, regularly interspaced, short palindromic repeats. And this is a mouthful that you're not going to want to say in conversation, so I'll just say CRISPR-Cas9. So the CRISPR-Cas9 system has been really useful to research. And basically, there's 3 main components. We need to have target DNA. So that's shown right here. And the DNA is double-stranded. We also need to have a guide RNA. And the guide RNA has a sequence that the researcher can design and pick in order to match up or base pair with the target DNA. So you need target DNA, guide RNA, and then the last major component is this Cas9 protein. And what Cas9 is going to do is find where the guide RNA and the DNA are matched up, and it's going to cut both strands of the DNA. All right, so this is what happens with CRISPR. Once the DNA is cut, the cell takes over. So when the DNA is cut, it forms something called a double-stranded break. So that's just both strands getting cut. And then what the cell will typically do is it doesn't want broken up DNA, is it's going to repair the DNA. So there's 2 different ways it can repair that DNA. It can stick it back together. So that's what's happening here. But this can be kind of a messy process. Sometimes the DNA gets stuck back together, but something gets kind of displaced. It's not a real precise process. And so many scientists use this process to generate what's called a knockout, or just a deletion. So if you want to just totally shut something down, like a gene, you can use this system. But the one that we're going to use is called homologous recombination repair. And what happens here is we've got our double-stranded break. However, this time, the researcher will provide a template. And so what this template is it's identical DNA on this side and this side to here and here. So it's identical to the location that's getting edited. However, this red box here is representing something that we want to put into that location. So we can actually cut and paste something and put it into that location where there was that double-stranded break. So this is the process that we're targeting, and I'm going to go through how we used it. So my whole first year of graduate school was the project of basically replace the PPK1 gene, the pickpocket 1 gene, with a docking site so we can do future editing. So here's how I did it. So the PPK1 gene is represented here just as a single strand for simplicity. Here's the PPK1 gene, and the yellow is just DNA on both sides of the PPK1 gene. So what I did was picked 2 sites outside of the gene that I wanted to make the cuts at. I then designed guide RNAs, and these guide RNAs are short sequences of RNA that are going to bind to the DNA through base pairs. We then introduced the Cas9 protein, and Cas9 is going to make the cuts. So we're going to generate that double-stranded break in the location that I specifically chose. Okay, the next thing I do is provide a template. So here is the template that I provided in the form of a plasmid. And the template has the thing I want inserted, which is this docking site, along with, you'll notice this yellow section. This yellow section matches up to the regions flanking the pickpocket gene. Okay, so then the repair we're targeting is called homologous recombination. So what's going to happen here is these regions are going to match up, and basically repair will go like this, and it will insert the item that I want into that location. And so the end goal is this docking site basically inserted into the site where PPK1 once was. And to confirm this, and these pictures are a little bit dim, but I think you should be able to see that in this situation, so this fruit fly larvae, you can see the PPK1, and this is again using that antibody marker, so it's a dissected larvae. So you can see it here, but down here, there's no PPK1. And so the PPK1 gene has been removed, and it now contains this docking site. And you may say, okay, well, why did you remove PPK1 if you want to study it? This CRISPR process takes months, so it takes a really long time to get from here to here. However, once we're here, we can use this fly to then knock things back into that docking site fairly rapidly. So we can knock in designer genes. For example, I can put back in PPK1 tagged with GFP, or green fluorescent protein. And so some of you may have heard of green fluorescent protein. This is what it looks like as a protein. It forms a barrel shape. And it was actually first discovered in jellyfish. So it's what allowed these jellyfish to fluoresce in the ocean. And so green fluorescent protein, by putting this onto PPK1, it allows us to basically see the PPK1 glowing in the larvae. All right, so this is the live imaging that we do now. So using that fly that I made with PPK1 tagged with GFP, I can then take a cover slip and put a larvae on that cover slip. I then put a piece of cover glass over top of the larvae. So if I rotate this 90 degrees, you'll see that there's a cover slip, a larvae, and then a cover glass. So this is a larvae that I take right out of the food. I rinse it off. I put it in between these 2 pieces of glass. And then I can image it with a confocal microscope and then see an entire animal and see what's going on inside the animal while it's still alive. And so you might wonder, well, is the larvae alive still after you do all this imaging? And the answer is yes. We can actually take the larvae back off, put it back in the food, and it's just fine. So we can image what's going on inside of the animal while it's still living. And so here's a picture, the very first picture I actually took when I imaged this for the first time. So this is showing PPK1 tagged with GFP. And you'll notice that we can see everything. So we can see every location it is in these neurons. So you'll see that the black signal, which is the PPK1 signal, shows up in all of these dendrites. It's in the cell body. It's even very faintly in the axon here. And so this was the first time we were able to actually see this protein in a live animal. We didn't have to dissect the animal or use antibodies. We could actually tag it with GFP. And so the other advantage of doing this, I'm showing you a static picture here, but we can also take videos too. And so this is a video of a section of dendrite. And you may notice, if you look closely, these little white dots, so watch this one, it's going to move. Or there's a little dot here where this arrow is pointing to, and it's going to move. And sometimes it's hard to see in the video. You got to kind of get used to seeing these videos. So I also represented these as something called a kymograph. So this is where time is on the y-axis, distance is on the x-axis. So basically, these white trajectories you're seeing are a particle of that PPK1 protein tagged with GFP moving. And does anyone remember what might be moving this around in cells? Close. These are the ion channels that are tagged, and it's the motors, yeah. So the motors are what's going to be driving things around like cars. And so the reason we can see this is because PPK1 is tagged with the GFP. So that's the ion channel tagged with GFP. And they move around, and they're moving because they're probably attached to a motor protein. So what we can learn from this is we can learn that PPK1 can travel on a motor protein, and that's part of how it gets to its destination. And so we're still doing a lot of research figuring out which motor proteins and how fast they go, but this is something we can do with our tagged PPK1. Another thing is we're going back to the Fragile X story. So I told you earlier that the Fragile X protein, when there's a loss of that protein in the fruit fly, less PPK1 makes it to the cell membrane. So I did an experiment where I took the PPK1 that I had tagged with GFP, and so this is going to tell us the total PPK1 in the cell. When we look at that GFP signal, it tells us a total PPK1. And so I'm going to then take a ratio of the total PPK1, which is the GFP signal, and I'm going to ratio it to the membrane PPK1. So this is, again, that marker. So this marker is labeling the membrane inserted ion channel, whereas the GFP is labeling just all the ion channel that's there. So we can basically compare the membrane inserted channel to what's just totally there in the cell. Okay? And I'm going to show this in a picture again too. When we take this ratio, we see that there's a decrease when there's no Fragile X protein. And so I'm going to show this in picture form. So in a control, when we're looking at the membrane compared to the total PPK1, the control has quite a few PPK1 channels in the membrane. However, we see a reduction in this when there's no Fragile X protein. So we actually see that there are a lot less of these channels inserted into the membrane compared to the total number of channels that are there in the cell. Okay, and you might remember from earlier, these ion channels need to be up here in order for an organism to feel pain, right? So the problem that's happening here with these channels not making it to the membrane is they're not going to be able to function if they're not in the membrane. And so to summarize what I just told you, Fragile X protein regulates the pain-sensing ion channel PPK1. So in control animals where Fragile X protein is present, it can regulate the process of getting these ion channels out to where they need to be to function, so in the cell membrane. However, when there's no Fragile X protein, this process is diminished, so less PPK1 ion channel is going to make it into the cell membrane. And thinking about this from more of a broad perspective, when we injure ourselves, such as with a hammer, and we feel a painful stimuli, our sensory neurons need to sense that stimuli. So the sensory neurons have ion channels. And what I've just told you is that we're studying ion channels and trying to figure out how ion channels get to the membrane in order to feel painful stimuli. And what we've discovered in our research so far is that the Fragile X protein actually regulates this process. So you need the Fragile X protein in order to get ion channels to the membrane. And this is really interesting when thinking about Fragile X syndrome. So in Fragile X syndrome, remember, this protein is gone. And so could it be possible that when there's no Fragile X protein and less ion channels are making it to the membrane, this plays a role in some of those sensory defects that occur in Fragile X syndrome. And so I hope you take away from this talk 4 things. And those 4 things are, what is a sensory neuron? So a sensory neuron allows an organism to perceive its environment. Additionally, neurons are highly specialized. So neurons are all one cell. So 1 neuron equals 1 cell, but that 1 cell has to have many different responsibilities, such as dendrites, which are going to receive signals, and an axon, which is going to send a signal off to the brain. Additionally, Drosophila, I'm biased because I work with them every day, but they're a great model organism to study sensory neurons. And so we can actually use gene editing techniques such as CRISPR in these fruit flies in order to study things in living animals. So we can really draw physiologically relevant conclusions because it's a living animal that we're studying. And then finally, the Fragile X protein regulates the membrane expression of a pain-sensing ion channel called pickpocket 1. And so we're starting to follow up some of this research, actually doing some pain assays on the fruit flies by poking them and seeing if this lack of PPK1 in the membrane in a Fragile X mutant actually causes the larvae to feel less pain. So we're literally doing that right now in the lab. And so I should have some updates on that in a few weeks. So with that, I want to thank my lab. So Jill Wildonger is my advisor. She's been a really great mentor for the last 3 years. I work with an undergraduate student in the lab named Jessica, and she's been awesome. Additionally, I want to thank my thesis committee for all their feedback on the project and our funding from the National Institutes of Health. And most of all, thank you to you guys for coming and listening, and I would be happy to take any of your questions. All right, I think we have a microphone, and then there's a question back there.
Yeah, so we want to get the questions on video here, so we need to pass the mic around as we go around.
What did you do with the PPK1 that you took out of the gene sequence? Did you just throw it away, or did you?
Yep, basically it just gets degraded. So when we're doing all of this, we're injecting things into Drosophila embryos. And so what happens is the PPK1 gets removed, and it just gets degraded by the cell. And then the thing that we want in there gets fixed, basically, and pops in there. So we're kind of, in a way, tricking the cell. So instead of letting the cell repair itself normally, we're actually making that cut and then putting something into that spot before it can fix it. And so the other stuff will just get degraded. Good question.
All right. So for the CRISPR gene editing, you mentioned designer RNA.
Yep.
I was just curious if you could comment on what designer RNA is, or how it's made, or how you match it to the target site.
Great question. So the RNA, the guide RNAs that we're using are pretty short in sequence. They're anywhere from 20 to 30 nucleotides. So remember A's, G's, C's, and T's. So we can actually design those. We can order one through a company. So they'll synthesize the guide RNA. And then what we do in the lab is we use cloning techniques to basically put that guide RNA into the larger guide RNA. So there's only one piece of the guide RNA, that 20 to 30 nucleotides, that's going to match up with the DNA. Then there's a whole other part of the RNA, which, let me point this out on my slide, that's going to let the Cas9 protein recognize it. So there's this part of the guide RNA, which we can control. That's what sticks to the target DNA. And then there's this whole other part of the guide RNA that just stays the same. So we do this cloning in the lab to basically make that. And then we inject it into the fruit fly embryos. Does that answer your question? All right, great.
When choosing to use GFP, how often does it fail, or is it one of those things that once you actually get the synthetic RNA set up, is it is it going to succeed, or does it fail multiple times before you can get it to succeed?
That's a great question. So the reason we chose the technique we did is because it has a pretty high success rate in fruit flies. So what I showed you here is basically we replaced PPK1 with this docking site. So this process is the process that took me like months to do. So getting to this point where I basically got a fruit fly, and remember the fruit flies are constantly making more of themselves. They're breeding. So once I make this in a fruit fly, I can, in a sense, keep it alive or make lots more of it by fruit flies just breeding, right? So once I've got this docking site, this is actually much more straightforward to then create something like a knock-in. So this step is quite straightforward. The first time I did this, it actually didn't work. I had to try it twice, and only 2 flies out of 200 that were injected worked. So the success rates for this step and actually doing CRISPR editing is still fairly low. I mean, it's amazing we can do it, but there's still a lot of optimizing that needs to be done. But the reason we put this docking site in is now I could even tag PPK1 with red fluorescent protein if I wanted to, or I could even make a mutant. So I actually have made some mutants of PPK1 where I changed the sequence of the protein and then put that mutant back into this location and study what happens in the fly. So the process of going from here to here in the fruit flies is more like 2 or 3 weeks versus like 5 or 6 months, and then if it doesn't work, trying again sort of thing. So yeah, that's a good question, because even though I told you fruit flies are pretty quick with their generation, like in 10 days you can go from mating the fruit flies to having more fruit flies, it still takes time to do these experiments, and so we want something that's going to be fairly rapid and successful. Yep.
So that GFP protein is covalently connected to your PPK1?
It is,
okay.
yep.
So how do you know it's not affecting the function or the ability for it to be motor transported? How do you
Yep.
know it's not blocking a site that it might bind
Yeah.
or something like that?
That's a great question too. So we're still characterizing this. However, I've done some experiments, and kind of like I showed you, where we're looking at the GFP tag PPK1 and we're using the antibody at the same time. So I can do this experiment to study what I was telling you about Fragile X, but I can also do it just with regular flies and then flies with the GFP tag and see if anything affects the membrane expression. And so this is a good readout to us to know whether or not trafficking by these motor proteins is disrupted. And so when I do these experiments, the GFP tag has not been affecting the levels of membrane PPK1. And so this is a good readout for that. The other way that we can figure out whether or not GFP is causing harm to the protein's function is we can do those pain assays. And so we're in the midst of really developing those in our lab right now, but when you poke the larvae, they'll roll away because they feel that pain. And so what we can do is poke just a regular control larvae, and then we can also poke a larvae that is expressing that PPK1 GFP, and they should both feel the same amount of pain based on the PPK1 making it to the membrane. We're still working on that. So that'll be kind of like a second check, but we have done this experiment, which we do see the same membrane expression with the tag protein. Yeah, thanks.
I was wondering, first of all, why was the name pickpocket given to that ion channel?
I've been wondering the same thing. So fruit flies, whenever you discover a gene in a fruit fly, you as a scientist that discovered it can name it. So there are some crazy names in fruit flies. There's a gene called hedgehog. There's a gene called Zelda. There's a gene called, let's see, what are some other fun ones? Oh, paralytic is one gene. It makes the flies paralyzed and have kind of like a seizure phenotype. So there's one called wingless. The flies don't have wings. So the pickpocket name, though, I've searched. I have not figured it out. There are other ion channels that people have discovered that when flies don't have these ion channels, they can't feel pain. And one of them they named Balboa, like Rocky Balboa, because he's like can't feel pain because he's a fighter. So I'm not sure who came up with pickpocket.
Okay, thank you.
Yeah.
I also have another question. So for those pickpocket ion channels, are those integrated proteins, and what makes them slow down not to make to the cell membrane, you know?
Yeah, what was
Have
the
you-
first part? Are they what kind of proteins?
So what kind of protein? Are these integrated that's ion channels, you know, the pickpocket ones?
Yep.
What? Membrane, are those
Oh, okay,
peripheral?
so they
and then, why you were saying that some of them make to the membrane?
Yep.
Some of them don't, and I was just wondering what makes them make it to the membrane, what makes them not make it to the membrane, what slows them down, kind of thing, you know?
You
That's
should
all.
come work with me, because those are all the questions I have. So this is what we're really trying to figure out. We're trying to figure out what makes pickpocket get to where it needs to be. So how does it get from in here to here? That's not well known in the field right now. So Fragile X plays a role in that, the Fragile X protein. We've figured that out. But there's going to be a lot more that we need to learn in order to fill this gap in, because there's a lot that has to happen from protein being inside the cell to being, I think you were saying integral membrane protein. Yeah, so from being an integral membrane protein, it has to be, so most everyone probably knows, remember the cell membrane is a lipid bilayer, so it's got 2 layers of lipids, and these ion channels have to poke through both layers, so they have to be a doorway through the membrane. So there's a lot that needs to get it here, and then one thing I didn't even mention is ion channels don't just stay on the membrane forever. We actually know that ion channels can then be recycled from the membrane. So this happens a lot in neurons that contribute to learning and memory. So oftentimes ion channels will be put into the membrane, and then they'll actually be recycled from the membrane, depending on the memory or whatever's happening inside the neuron at that time. You're welcome. Yeah.
Tim had to leave, so I'm
Okay.
moving this around.
Is there any way of knowing, or is there any knowledge regarding the time scale at which the proteins are developed and then get to the membrane?
That's a great question, too.
If we don't know, could it be just, I mean, I wouldn't assume this, but probabilistic in that they make it to the membrane? I mean, how do we know that they're driven there?
Yep, so how do we know that they're transported there? So we're currently doing some experiments to figure that out. And those experiments involve, let me show you this picture. Okay, so when we're imaging this, what we can actually do is use a technique called FRAP. It's fluorescence recovery after photobleaching. So what that involves is we take a laser and we shine the laser just on a part of the dendrite. And what it's going to do is it will bleach those fluorophores. And so the GFP is shining bright, right? But when you actually bleach it, it's going to turn off. So we can turn off the GFP in that little section. The rest of the dendrites look fine. And then what we watch for is in that little section that we bleached, we watch for recovery, basically. So does anything come back in and fill in that gap that we made? And so we can start to look at your question, which is time scale, which is super important. So we're trying to still figure that out, but it seems like the pickpocket is delivered to its site of location pretty slowly. So it's not something where it's just happening real fast, but it does seem to be transported by motor proteins to get there. So that was shown in that video, so we can actually see the pickpocket protein moving. So it's delivered by motors, but we're still trying to figure out which motors it is, how fast those motors are going. So there's a lot of questions. Just like any scientific question, there's always more that you're wondering, but we're going to figure it out. Thank you for asking that. Any other questions? Yeah.
Are there any, because it sounds like we do understand motor proteins, at least to an extent, so
Yep.
how do we know of any motor proteins and their associated ion channels, and like, are there some of those processes that are just pretty well understood generally that we can
Yep.
take reference from and learn from?
Yeah, so we are learning a lot about, so there's 2 major motor proteins. One's kinesin and one is dynein. And so these motor proteins are basically driving in opposite directions on the highway. So one carries cargo this way in the cell, one carries cargo this way. So dynein does one direction, kinesin does the other direction. We do know some cargos. So we know that kinesin carries a certain cargo to the axon, or dynein carries a certain cargo to the dendrites. There's still not good examples of ion channels that are localized in the dendrites and what motors transport them. We know a lot more about what's going on at the axon, like the synapse location, because that's studied a lot in like learning and memory. So people are looking at what's happening when a neuron synapses on another neuron. What we're looking at is kind of this first line of defense. So it's the very first stimulus that's happening to the neuron, and then that will then get sent on through the circuit up to the brain. So yes, there are a lot of examples, but really not any examples of ion channels that are transported out to these dendrites. But we are trying, like, of course, you read the literature and you try to get an idea of examples that are happening in other systems and then figure out if it's happening in yours. So we're in that process. Yeah. Yeah.
If Fragile X can change how many are expressed in the membrane, is that acting as a motor protein, or is it a different way that it's acting upon it? And then if you were to just get rid of that and then do the bleach test, would that like change anything?
Mm-hmm.
But
So yeah, good question. So that is an experiment that we're doing currently too. We're doing that FRAP or bleaching test in the Fragile X mutant. Is Fragile X a motor or acting as a motor? It's not. So it's not a motor protein itself. It has been found to act as an adapter or a linker between a motor and an ion channel cargo. So we're starting to investigate whether or not it's actually acting as a linker between motor proteins. The Fragile X protein's most well-known job is it regulates translation. So Fragile X can bind mRNA, so not even proteins. So there could be some regulation happening at the level of making mRNA into protein. And then the third thing that Fragile X can do is it can bind ion channels directly. So there is an example of an ion channel that is known to interact with the Fragile X protein. And what Fragile X does there is it actually recycles the channel from the membrane. So it's part of the process of taking the channel off the membrane. So we're trying to see kind of those three options. Our hypotheses are something's going on in one of those three scenarios with the Fragile X protein, but we're still doing experiments to narrow down what that is. Great question.
In the interest of time, please join me again. I thought that was phenomenal.
Thank you.
Thank you, Josie.
Thank you so much for coming, guys.